He's MD, PhD, and he's a associate professor of medicine and immunobiology at Yale University. He got a training from Harvard University and UT Southwestern, and also did a fellowship at Yale New Haven Hospital. So his research is about immunology and immunometabolism and immunity and pathophysiological condition. His talk is about the immune system and lowering adaptive physiology. Please welcome Dr. Wang.
Thank you, Sung-Jin, for that very nice introduction. Thank you for the organizers. It was very nice to be in Korea. Like Florent, I'll be talking to you about mostly physiology. As Sung-Jin mentioned, I'm also a medical doctor, and so I'm very interested in keeping people alive. And so as a result, my lab spends a lot of time trying to understand the biology of survival. And our approach is to actually think about, during evolution across species, the types of things in the environment that would drive specific kinds of adaptive programs to enable survival. So, for example, all animals will have to deal with not having water, not having food, getting chased by a lion, and so on. And what you'll notice is that with every specific kind of environmental threat, there is a corresponding and very particular kind of physiology that enables dealing with not having water, not having food, and they're different. The other point I want to make here is that the behavior adaptation is actually the solution to the problem in the environment. So in other words, you can, in Florent's example, you can do non-shivering thermogenesis as much as you want, but unless you find warmth, you're not going to fix this problem of the cold. So another way of thinking about this is that the physiology enables the behavior which solves the problem. So it turns out there's a lot that we don't understand about normal adaptive physiology in general, so I'll give you some examples. This example on predation we published a few years ago, and it came from clinical observation actually. So my mother has lupus, and what I noticed, many other doctors have noticed, is that when something stressful happens in their life, their inflammatory disease actually flares. And this is true for, here I'm giving you lupus, it's true for MS, it's true for many of our diseases. And it's very counterintuitive because the stress response is usually catecholamines and cortisol. And for the physicians in the audience, we use these as drugs to suppress the immune system. So what should happen is when they get stressed, their disease should get better, but that's not what happens. And so what we discovered was actually in a large screening campaign in mouse and in humans, that IL-6, which we normally as immunologists think of as a cytokine, is also a stress hormone. And so you can see, for example, this is after an acute stress. Here's adrenaline, catecholamine, and you can see IL-6 come up here. And this induction of IL-6 with stress requires that the mouse is awake. This was beautiful work done by Reina, who's a PhD student now at Yale. She was a postdoc at the time, and Huaqing, who's now a lead scientist at Genentech. But the basic idea here is that IL-6 is induced to allow the animal to be able to run away. This is what we showed. And since it also enhances inflammation, one can imagine how this would increase inflammatory response. It makes a good deal of sense. Usually if you're running away from a lion, you probably will get cut or get bitten. So you would want to actually anticipate a good inflammatory response. I think it's very interesting because this finding actually matches a lot with genetic studies and also observations in the psychiatry field, where that depression and anxiety, folks with this diagnosis, you can often find IL-6 elevated in their serum. And in fact, there's a clinical trial ongoing now testing the effect of IL-6 blockade in treating depression and anxiety. And so the general question here then, from a kind of a psychiatric disease perspective, there has been this idea that there is an inflammatory component to this, and this is the idea that the immune system is really involved in maintaining physiology. So there's been this lingering question of what is the role of the immune system in modulating kind of the homeostasis of the brain. And those of you in the audience know that essentially in any brain disease, and so I have a couple of examples of here, from Parkinson's disease to Alzheimer's to multiple sclerosis, where the disease is correlated with finding immune cells in the brain parenchyma, which they really shouldn't be in, right? And the question has always been, is this bad? I'll give you a very, very recent example that was not published by our group, but I found fascinating. And it was published in a relatively specialized journal. And so clinically, we use tau to diagnose Alzheimer's. And so you can see that in age-matched patients, Alzheimer's have more tau. This is in blood. It's true also in CSF. They thought to measure it in newborns. Look how much tau is here. So saying that tau is pathogenic clearly is not entirely correct. Turns out other species, hummingbirds, for example, have a ton of tau and they fly around just fine. Okay, so we sought to tackle this. I'm going to go through this fairly quickly because we published this recently, but the idea that the brain is immune privileged comes from very old experiments done with xenotransplantation by Medawar, who won a Nobel Prize for this. And what we knew, because I live on a farm actually, this is my youngest daughter, it was known actually when Max Cooper discovered B cells that there are B cells in circumventricular organs in the chicken where B cells were discovered. The B stands for bursa fabricio, which is a chicken organ. And this was published in 1980. And so other vertebrates, there's already an example in other species where they have lymphocytes anatomically focused in brain parenchyma. So we asked the question, is this true in mammalian brain? And this was beautiful work done by a PhD student, Tomomi, who's now a postdoc in Carl Deseroth's lab, and done in collaboration with David Hafler. And so what we sought out to do was basically, we generated a mouse, and I'm showing this video for a couple of reasons, where CD4 cells are lit up with tomato, and then we clear the whole brain, and then we do light sheet microscopy and 3D reconstruction of the brain. And you'll notice immediately that this is not lighting up just cells that have that are this right size as lymphocytes. Now I'll pause the video when it goes into the 3D rendering, but you can see that it's stereotypically labeling actually nerves. And this is a constitutive CD4 promoter. So for immunologists, we use CD4 often to delete things from T cells, but for example, this lights up pretty specifically the optic tract. And this is what the constitutive CD4 is doing. And so this was a big surprise to us. And so since then, we've worked with Sasha Densky's group, and we actually generated a version of this where we can make sure that we have better control over this, because clearly the CD4 promoter is accessible and development. All right, so what you can do then with a 3D map is you can plot the density of cells, that's what's on the y-axis, against all of the locations of the brain by mapping it onto the Allen Brain Atlas. And then in an unbiased way, say, where do I get the most TD tomato signal? And you can see the subcortical organ popping up in unbiased analysis. And we were actually not that surprised to find this, because the subcortical organ, like the chicken pineal gland, is a circumventricular organ. And then we, using autopsy samples in patients that didn't have brain disease, so we collected these 2 hours after death from the Yale Rapid Autopsy Program, we found the same thing in humans, that there is a concentration of CD4 and CD8 T cells in the subcortical organ. So the obvious exercise that we're pursuing then is this atlasing of the number, the anatomic location, the quality, the phenotype of lymphocytes in brain parenchyma across health span and across various disease states. I'll give you one example of this, which we haven't published, but this is a model of early life stress. So basically you separate the baby from the mom, and then you socially isolate it, and then this is a common model used for early life trauma. And what you can see is that there's a great increase in the number of T cells. This didn't project very well, I apologize for something happened in the conversion. Great number of increase in the T cells, and unlike T cells at homeostasis, which are primarily gamma, they now make a lot of 17A. So this is fairly consistent with work from Gloria and June at Harvard and has this kind of feeling of their findings in autism. I think the main question again is this bad? It's not entirely clear. Okay, so the one physiology behavior match that hasn't totally fit and has kind of been a problem in the field has been actually with infection, right? So it's not obvious that sickness behavior, unlike finding water, finding food, is actually something that fixes the problem of infection. And so sickness behavior is very, very stereotypic, right? It includes these alterations in motivational behaviors. And we know that the immune system plays a role in this because it's very clear that immune signals modulate feeding centers. So both feeding centers in the brain, in the arcuate nucleus, the hypothalamus, which kind of deals with normal kind of satiety, and also this so-called emergency part of the brain in the AP and MTS. This is incidentally where GLP-1 drugs act. In the area postrema in the nucleus of the tracts solitarius. And this is some work that we've done in this particular space. But the point here is that there is definitely immune control of appetite. Okay, it also makes sense, these parts of the brain make sense because the blood-brain barrier here is actually not totally together. And so these parts of the brain can sample peripheral blood. So they can be connected to the events happening in the rest of the body. Okay. This is really, really medically important. So all I've done here is collect over the number of clinical trials over the last 7 years on what should we feed patients when they're sick. So these are patients that can't pick themselves. And the short answer is we still don't know, which is very, very surprising in 2025. And so what I'm going to show you next is data that's not published. And the question that we wanted to ask is if there is such a thing as sickness behavior in which we know that appetite is modulated, how about in recovery? Is there a corresponding recovery behavior in which there would be stereotypical changes in appetite? So that's the last part of the talk, and I hope you find it interesting. This is work done by Nikolai Yashka. This is a shameless recruiting for him. He just started his lab at University of Hamburg. He's recruiting all kinds of people, and it was done in collaboration with another postdoc who's a neuroscientist and psychiatrist. Okay, so we asked a very simple question, which is, after sickness, and we modeled this with one low dose of LPS where they don't eat for 24 hours, or we took food away for 24 hours. So this is true in both cases. I'm not presenting all the data, but just giving you a high level. And we let them pick, do they want to eat food that's high fat, high carb, or high protein? These are isocaloric matched, micronutrient matched, everything matched. And this is spontaneous, so they get to choose. So what you'll see here is that they don't really like to eat the high protein diet. And since it's isocaloric, they then don't actually eat as many calories. Okay, so it seemed to us that they didn't like this food. So Nikolai asked a very interesting question, which is, what if we force them to eat it? We create an environment in which they have to eat it. And so he played this trick with a small animal. If you put it in the cold, it has to eat more to survive. So this is what you see here. So this is a high-fat diet. So this is at 30 degrees. And then after recovery, you put it in the cold, you see they eat about 50% more. And this is how they maintain survival. If all they have to eat is high-protein diet, they don't eat more, and they die. So this is they have free access to the food. It's just sitting there, and yet they won't eat it. And this is unique to the recovery state. All right. So if you have a healthy mouse, they'll eat it. If you have a recovery mouse, they won't. Okay. So this seemed a lot like aversion, and so aversion is encoded in the APNTS, the same place as drug target for GLP-1. And so the question is it on? And the answer is yes. So we did this a couple of ways. One way is by gavaging them the same number of calories of sugar, fat, or protein, and then look for neuronal activation by phosphatating in the area postrema and the nucleus of the tracts solitarius. And you can clearly see that there's a lot more neuronal activation with protein. You can also see this if you just put the food in there and let them eat it, and then do see phosphatating after ad libitum consumption for about an hour. So what the brain is monitoring here is not calories, it's monitoring what calorie, which is very, very interesting. Turns out that a couple of clinical trials were done in the last 3 years actually asking the question in recovering patients, should we be feeding them high protein diets? Because this is what we currently do. This is the first time these clinical trials have been done. We've been doing this for the last 50 years, where we reason that patients lose a lot of muscle, so we should feed them high protein diet. But when you do the randomized control trial, it turns out that not only is there no benefit compared to a normal protein diet, there's a signal for harm. And this was repeated in 3 separate randomized control multi-center clinical trials. Okay, so how is this working? We wanted to understand what's the mechanism of aversion. So a lot is known about this, from how you smell the food, to how it stretches your stomach, to the neuroendocrine hormones that are released after you eat it. This is beautiful work done by Zach Knight, Brad Lowell, and many others. We tested all of them, and we were unable to find canonical mechanisms of aversion to protein. So we were stuck actually for a while. So we came up with this interesting hypothesis that maybe there was something intrinsic in the protein that was aversive. And so we reasoned that for it to be aversive, it probably had to be toxic. And so we did actually a screen in mice where we took the model protein, which is casein, and we basically put each proteinogenic amino acid, so 20 of them, just like in casein, and gave them in single bolus, all 20 of them into mice. We did it in a non-physiologic way where we gave the entire amount that they would eat over 24 hours all at once, and we were screening for toxicity. And so this is the data. So you can see 17 of them, the animals do fine. They will eat high-fat diet after recovery, but in 3 of them, actually, we killed the mice. And so this is glutamine, lysine, and threonine. This was a very surprising finding. Obviously, this is not physiologic because we're bolusing the entire amount that they would normally eat over 24 hours all at once, but this was meant as a screen, okay? So then we generated diet where the diet lacks these 3 amino acids, and in this diet, we increased the other 17 proportionally so that still calories are matched. And then we just let them pick. Do you want to eat the one that without the 3 amino acids or the one with the 3 amino acids? You can see in the fed state, they have no preference. But now in the fasted or post-LPS state, now they greatly prefer the isocaloric diet that lacks these 3 amino acids. So this is completely physiologic because they have free choice of which one they want to eat. This is also true when you put them in the cold. And then because they'll eat it in the cold, then unlike the amino acid diet, now they survive the cold because they eat. So we had tried to figure out what the mechanism of toxicity was. And I'll give the kind of spoiler alert here. It's not totally satisfying because there's a key part of this that we don't understand. But this is the background. So when you metabolize amino acids, the first step is the production of ammonia. And so ammonia, as you know, is toxic. And so the liver then detoxifies ammonia into urea nitrogen, which then you pee out. Okay, so this is the normal kind of detoxification of protein. So we asked the obvious question, are QK and T more ammoniogenic? And so either if you put the amount that's in casein back to the animal, or if you give them all in the same amount and you match them with biochemically similar amino acids in vivo, each one of these amino acids, QK and T, is more ammoniogenic than biochemically matched amino acids, both if you match the amount or if you put the amount back that they would eat in casein. And we don't understand the mechanism for why this is. This is very, very confusing because the first step for the metabolism of amino acids is the release of ammonia. So it's not intuitive why this is the case. But we're pretty sure it's ammonia, so you can do this trick. For example, this is a drug that's used in cancer where you block conversion of glutamine to glutamate, so you don't release the ammonia. And when you do this, then glutamine no longer kills. You can do this also trick where mammals can't use the D enantiomer, so they can only metabolize the L enantiomer. So you can't make ammonium this way. And then also ammonia is sufficient. So if you give increasing amounts of ammonium acetate, compared to an osmolar controlled sodium acetate, you can in a dose-dependent way kill the animals. You can also create aversion, right? So if you pair a novel flavor, grape, with ammonia, then reintroduce it, and you let them pick. Do they like the one where they experienced the toxicity of ammonia when they first met grape? Now they don't like grape, and the same is not true with the isoosmolar control. And you can gavage them some ammonia, and then they won't want to eat the high-fat diet in the cold, or you can put it in the drinking water, and they don't want to eat the high-fat diet. So ammonia is intrinsically aversive and causes anorexia. Okay, so if everything I'm telling you is true, then ammonia should also light up the APNTS. And so we did this using a clever system, which is called the FOSS-TRAP system, where basically while the animal's eating protein in recovery, we give tamoxifen, and it traps the neurons. And so they're marked here, TDT, and then we rest the animal, and then we give it ammonia, and then we stain for CFOS, and then we look for co-localization. So this is a combination of a genetic and immunofluorescence technique, and you can see that there are a high percentage of neurons in the APNTS that are responsive to both protein and ammonia, suggesting that they're sensing the same thing. Okay. The last part of the talk will be where is ammonia sensed? And so the first thing we did was, where is the amino acid metabolized? And so to do this, our lab uses a lot of radio tracing, so we did 14C glutamine tracing. So here the carbon is 14C, and then we give animal LPS, and then we gavage them 14C glutamine tracer amount, and then we look at all the organs. And you can see that the tracer is recovered pretty much primarily in the proximal GI tract and really in the duodenum, and really not in the brain. Right, so this is where the amino acid is metabolized. So where does the nitrogen from the ammonia come from? So to do this, we made a 15N glutamine, and then we did tracing and then mass spec looking for where is this 15N in ammonia, right? So the 15N comes from glutamine, and we're looking for the 15N in ammonium, and you can see that we can recover it in the same place where the amino acids are being metabolized. And for a sanity check, we can also see it incorporated in urea in the liver. Okay, not to present all the data, so there's some time for questions because I'm at the 20-minute mark here. To make a very long story short, Artem from David Julius's lab had shown that TRPA1 was a ammonia sensor. So here we took enterochromatin cells, which are the only cells in the proximal gut that express both the enzymatic machinery for making serotonin and also TRPA1, which is the ammonia sensor that was described some time ago. So we ex vivo isolated enterochromatin cells in a TRPA1 sufficient and lacking context, and then did electrophysiology. And so you can see ammonium clearly causes depolarization of the enterochromatin cell, not the osmolar control, and here's the positive depolarization control. And this does not happen in the absence of TRPA1. And then finally, we generated a TRPA1 conditional knockout. So this is using chromogranin A, CRE-ERT2, TRPA1 flox. We got some help from Kevin Tracy and Charles Zucker for this. And again, you can clearly see here that when TRPA1 is missing just from enterochromatin cell, where the ammonia is being made and the amino acids being metabolized, now you have much less C-phos activation. They eat in the cold and they survive. Okay, so what we think we have discovered here is a specific recovery mechanism to prevent protein toxicity. So when you think about when you're sick, you may not smell as well, you may not taste as well. Your GI tract doesn't move normally, so this is a condition called ileus, especially in infection, the GI tract doesn't move. And then many of the neuroendocrine hormones that are made during inflammatory settings, like during infection, don't have the same meaning, right? So insulin may not mean the same thing when you're inflamed and when you're not. Growth hormone is another good example of this. We think that since these things probably may not operate, there is a backup system, kind of a last resort system, which relies on real-time sensing of amino acid metabolism at the level of absorption and measurement of how fast this is coming in, and then stopping you from eating. So this is the model, right? So there is this systemic detoxification, which I showed you, that's been known, but this is too slow, right? Because by the time you're sensing ammonia in the blood, ammonia is toxic. So you want an anticipatory mechanism here to stop you from eating more food as it's coming in. This is exactly the same design as GLP-1, right? If you wait until there's high amounts of glucose in the blood for beta cell to make insulin, it's too late. So this system anticipates the glucose load. And if this is true for protein and this is true for sugar, this must be true for fat and for many other things. So I think in addition to this kind of canonical idea of physiology and behavior, we now have another category of, is it possible that there are actually dedicated recovery states to each one of these kind of environmental threats? And I think that's probably very likely, because the kind of stress that this imposes on the system is very different in each one of these contexts. And then finally, we started working on this because of long COVID. It might be that diseases like long COVID are actually diseases of pathologic recovery. And long COVID is an example of this, but Lyme disease is another example of this. Incidentally, Lyme disease was discovered at Yale. This is actually generally true after critical illness. And this phenomenon has been described really for the last 100 years. And so this is kind of how we think about disease. We study what's normal and then try to understand what breaks to make disease. And with that, I'm happy to take any questions. This is my awesome lab. I'm very grateful for my mentorship, Ruslan, and my clinical mentor, Christina, and all of our funding. And I'm particularly grateful for Sungjin for the invitation and for the organizers for such a lovely event.
Thank you so much for a great talk. We will take questions from our floor.
Yeah, thank you for the fantastic talk. I have a question on the specific case of protein deprivation. So when the mice are provided with the foods deprived of amino acids, they prefer protein-rich foods, or even like twice or 3 times more consume protein-rich foods. So
yes.
how does this mechanism tolerate it?
Yes, so that's a great question. So what we describe here is the regulation of the upper ceiling. You're asking the question of the minimal threshold. What I didn't show is when they eat a lot of the high-fat diet after recovery, if you measure the amount of protein that they ate that's present in the high-fat diet or the high-carb diet, and you look at the total amount of protein eaten across all three diets, it's exactly the same. So this is what Steve Simpson described as protein leverage, where if a food is mostly fat and has a little bit of protein, you'll eat as much of it as you need to attain a minimal amount of protein. If the food is lacking in essential in amino acids, especially essential amino acids, then it has been proposed that GCN2, which senses uncharged tRNAs, in particular for essential amino acids, is the mechanism by which the organism knows it needs to find foods that have amino acids. But this data is a little bit messy, and we certainly were not able to reproduce this. But our study is really trying to establish the upper limit, not the lower thresholds.
So in that case, too, ammonia concentration is also like in the medium range.
Yeah. So what you'll see actually is urea nitrogen is different. So if you, for example, are on Atkins diet or you eat just steak, what happens actually is in the beginning. So eventually, what will happen is liver upregulates all of the enzymatic machinery for making urea, and you make more BUN and you pee more. So this is the acclimation. What we're describing here is adaptation. So in the same way that you go up a mountain, you breathe faster, but then when you live up on the mountain for a long time, you make more blood. It's a different strategy if you have to do it for a long time.
Very beautiful talk. So I may have a naive question. So for the ammonium, in healthy people without diseases, so what's the physiological function for the amino generated from those protein diets? That's the first one. The second one is we still prefer, for normal people, we're still able to prefer protein diet, right? So does that mean the neuron circuit level, whether this pathway will somehow inhibited, like by smelling, tasting pathways? Thank you.
Yeah. So for the first question, I'm not sure if you know what physiologic function of ammonia may be. It's an unavoidable biochemical step if you want to metabolize amino acids. The second question is about the kind of organismal context, and I think we demonstrated this. This matters a lot. And so I went over it a little bit quickly, but we tested smell, taste. So we used tasteless mice. We used mice where the stomach can't stretch. So this is a lot of work that's done kind of in homeostasis. I think what's interesting here is that in recovery, it relies on a different mechanism of sensing.
Yeah.
And so I think it's very interesting. I don't know, we'd have to test it in all the different kinds of organismal contexts.
My question is actually, in the normal conditions, whether this sensing pathway from ammonium to the brain is suppressed by the taste or smelling, those more enjoyable circuits.
It may not actually need to be utilized, right? Because in other words, if you're asking whether or not it's required even at steady state, I don't think so.
Okay, thank you.
Andrew, here. Fantastic, as always. Thank you.
Just the last question, sir.
Okay, briefly. So, because you already showed that glucose has an opposite effect during like infection, depending on the type of insult. So, have you looked at the different type of stressors in your recovery model?
We have not, but Nikolai plans to do this basically systematically.
Thank you.
Thank you very much.
Okay, it's okay.
The next speaker is Dr. Hyung-Pyo Kim.